inheritance. All right, so seeing that our topic is inheritance, that means that we are going to be studying more about genes. We're going to learn about why, children are born with different characteristics from their parents. We're going to learn what exactly takes place within the cell in order to allow for these differences to manifest themselves. Now, we're going to be introduced to a lot of terms, so I want you all to take proper note of the terms so that you will be able to keep up as well. And then we're going to take our time and go through the processes of cell division, because remember, you all learned about mitosis and meiosis before, but we're going to actually learn about what takes place in each of them and how, what goes on with it in each stage of cell division when it is you are speaking about both mitosis and meiosis. All right. If nobody got a chance to look at the videos that was sent, I will show them back. And I also have one additional video to show about, how you call it, meiosis. Like rip jeans. Yeah, actually, it's like ripping your jeans in a sense. Except jeans would be, in this case, these jeans. So yeah, you're not entirely wrong with that. So we're gonna learn about how it splits, how we get our characteristics, what determines what we see, all right, what we see physically, and all of that. Okay, so we're ready to start. Let me share the screen. All right. So a lot of y'all, well, Emily, this would look familiar to you, and it as it
Actually, in HSB students such as Jesse, it would look familiar to him too.
Right, exactly. So y'all should be well aware of this, so it should be easy. So then y'all should be answering the questions like firsthand. I noticed Dimitri isn't signed on, so the competition will then be between all of y'all. All right, even if Dimitri were here, okay? So so yeah, y'all would have a lot of stuff to answer. So this is what we're gonna learn, all right? Or review in the case of the HSV students. All right, we have to learn what is meant by inheritance. You're gonna have to understand as well what we mean by chromosomes, genes, and alleles. We're going to learn a little bit about protein synthesis, which we have spoken about before already, so it will just be like a review. All right, remember we went into detail talking about what goes on with your DNA and your RNA in order for you to get a particular protein, and then why is this unique? Why is this important for why is this an important process actually? Why do y'all think it's important? Y'all remember? Tell me. What does protein determine? Why is protein particularly, the type of protein particularly important to a cell? Let me hear you.
Because of the function, something to do with, I can't remember, miss. I tried.
Yeah, you're right. It has something to do with the function. So it is related to function. So depending on the type of protein that you have, that is the function. That will determine the cellular function. All right. And the type of protein that you have depends on how the amino acids are folded and what determines how the amino acids are folded. Like, how do we know this amino acid has to be here, that amino acid has to be there? How do we know all of that? Three, two.
No, miss, I can't remember. I can't remember.
What did I say? We just said it. Well, the specialized cell specialization, yes. But what determines all of that? Remember, what is it that determines how the amino acids are folded? What instructs the amino acids to be folded in a particular manner? The answer is
Protein?
no.
Yeah, I
No.
don't know, miss.
What is inside the nucleus, Dimitri, that will control that? That controls how amino acids are folded. Right, there we go, DNA. And the answer is right on your screen too. DNA and cellular function. Oh, sorry. Sorry, yeah, David, sorry. I see you. Right. Okay, just now, hold a sec, y'all. Okay, so we resume. Right. So as we were saying, your DNA is what is responsible for how the amino acids fold. And depending on how the amino acids fold and come together, that is going to determine the type of protein that you have. That type of protein that is formed will determine cellular function. All right, whether your cell is a cheek, a muscle cell, forms part of your nervous tissue, or like it could be anything, whether it be a gamete, whether it be um, a nail cell, hair cell, anything, basically. That is what is going to determine what type of cell it is. All right. So that is what we mean by, protein synthesis and how DNA affects us. So always remember that, okay? We're going to learn about mitosis and meiosis predominantly in this presentation, and then the next one, we go into actually calculate the possibilities of offsprings, all right? If you all had gotten the opportunity to review it, you would see a whole set of stuff about genetic diagrams and stuff. That is what we're going to use in order to determine possible offspring outcomes, all right? So yeah, we have some practical work ahead of us. So roll up your sleeves, tie up your hairs. Emily already did, so let's get started. All right, so we learned about chromosomes last time, right? Remember, we said that they are located in the nucleus of every cell. And we had also said that they cannot be seen unless they, unless the cell is starting to divide, they become short and fat, and that is actually the time in which you would get to see them, all right? So remember, we also looked at how DNA is wrapped around certain proteins. What are these proteins called, and how is it that you get the chromosomes? Tell me, talk to my people. How the chromosomes are formed. So we know that DNA is wrapped, but tell me, what is it? How is your DNA coiled on to form a chromosome? Who remembers? So we have our DNA, which is wrapped around certain proteins called, it starts with H. And there's a letter missing. Close. HIST is correct. So we have that HIST. Then we have, yes, histones. Yay, excellent. History, they're wrapped around history, David. Oh gosh. No. Well, they do have, remember, if you look at it from this standpoint, everything about you from your history to present, even your future, is all recorded in your genes, right? In your DNA. So, yeah, your DNA will carry a history, but they are wrapped around certain proteins called histones. And when they are wrapped around them, they form these little balls, and then eventually they coil and they tighten up, and then they will form what is called your chromosome, what you see as being your chromosome, right? So that is basically where your chromosomes are. And they are located in the nucleus of your cell. So they are found in the middle, all right? They are found within the nucleus of every cell. All right. And Basically, a gene, we hear about genes, we hear about DNA, all right? Well, we know DNA stands for
Deoxyribonucleic acid.
how I know Emily was going and say that. I feel like I could hear the excitement in your voice, like, ooh, I know this
It's like,
one.
yes, this is my time.
Very good. I'm glad. So shine. Right? So yeah, that is what DNA stands for. And what a gene is basically just part of that DNA molecule. So the DNA is this whole big molecule, and a gene is just part of it. And your genes have certain codes, all right, which basically is like a language that will, be translated in order for a protein to be formed a particular way. All right. Your genes are basically everything that you are. Everything that you are is recorded in your genes. Your eye color, hair color, every single thing. It is recorded in your genes. They determine all of your characteristics, basically. Not just you, but even your children, your children's children, and the children of your children's children. All right. Everything is all determined in your genes, and that's why you could do gene mapping or even ancestral lineage testing, and you could find out your ancestors. You could find out who you came from and which part of the globe you're from, your ancestors from, if you have Spanish, Jewish, French history, anything like that. It's pretty cool though. It's a vast world that scientists are still exploring, but it is pretty cool. Okay. All right, so remember we spoke about the amount of genes that your cell has, right? So how do you call a cell that has 46 chromosomes? It starts with D and it ends with D. It has a P in the middle. So it starts with D, it has a P in the middle, and it ends with D. A cell with 46 chromosomes.
A diploid. Oh, Dimitri just said it.
Very good, Dimitri and Emily. It's called a diploid cell because every cell in the human body is supposed to have 46 chromosomes. But which are the only cells in your body that don't have 46 chromosomes, and why? Sperm and, right, gametes. So yes, sperm and egg, otherwise known as gametes. Very good. Why do they only have 23 chromosomes though? Why they can't have 46? I mean, why do they have to be so different? Excellent. So when they combine, because remember, sperm has to meet egg. So when sperm meets egg and combines, the 23 plus 23 will give you a total of 46. Very good. So then if a dog, let's see, should I use, yeah. If a dog if a diploid cell in a dog has 38 chromosomes, how much would a haploid cell be? $38.
23.
No, remember, haploid would be half of 38, which is 19. Right? Remember, hap is like half. So all you have to do, no matter what number you have, if you have to say how much would a haploid cell contain, you just divide the total number by half. Right? So hence the reason it would be 19. Okay? Okay, let's do one more. If a diploid cell is 24, has 24 chromosomes, how much would a haploid cell have? Excellent, 12. Half of 24 is 12. Very good. Okay. So every cell in your body has an exact copy of all your genes, right? So that means that the same genes are throughout your entire body. All right. So genes for your hair that would be responsible for coding the protein that will be responsible for forming keratin, they have also found throughout your body, yeah? But it's not to say that you're going to have the type of hair that you will find on your head growing out of your toe. All right, one morning. That's not going to happen because that particular gene will only be activated in that part where your hair is supposed to grow. Right? Your body's so amazing, isn't it? Wow. All right. So yeah, but twins would have similar combination of genes, but yet it's different. All right. Hence the reason you would have so much of similar behavioral patterns, but it won't necessarily be, you know, real identical, right? Like even identical twins are still different, all right, genetically speaking. So yep. All right. So we know about haploid cells. We know that from fertilization, as soon as the sperm nucleus meets the egg nucleus, we have fertilization occurring and we have a zygote being formed. So remember this process that we had outlined, fertilization to zygote. Then after zygote comes, after zygote comes So from fertilization, you get a zygote forming. After you have a zygote, that is when the cells will continue to divide to form an embryo. And then the embryo is what will develop into a fetus. And the fetus develops into a big hardback human like ourselves, right? Um, so we know about diploid, we know about haploid, right? But now we get to homologous chromosomes. What exactly do we mean? We know about chromosomes, but what do we mean by homologous chromosomes? All right, it just basically refers to two chromosomes that comprise a pair of chromosomes, right? So homologous is just two parts of make up a pair. Remember, a pair is two. So homologous chromosomes is just basically two chromosomes of a pair, which I will show you just now in a picture. One single member of that pair is called a homolog, all right? So look at it here. All right, so these are the 46 chromosomes that make up a man. All right, it cells within a man. As you can see, the X and the Y chromosome here. So this is the sex gene. All right, but notice each of these, you have two of them. So these are all called homologous pairs. If you just had one of them, that is called a homolog. All right, one will just be a homolog. Two of them will be homologous pairs. And because we have two of them in each, all right, we have 46 chromosomes total. So notice how they labeled it here for you. One, this is 22, and the sex chromosome is 23. So 23 by 2, because each of them have 2. So if you count them, you will get a total number of 46, which means that these are the chromosomes of a diploid cell. Therefore, a haploid cell will only have one of each kind of chromosome. One of each kind, that's all it will have. And it will divide to produce that same number of one of each kind of chromosome. You all understand that concept?
Yes, miss.
Wonderful. All right. So this is for a man and this is for a woman. All right. These are all homologous pairs as well. Again, one of each will be a homolog. And this here, the final gene is your sex gene. All right. So that's the reason all of your cells have all of these chromosomes in them. all of them. All right. Now, when it is, yeah, like let's just say, for example, you take a cell sample from an arm and you don't know if it's a man or a woman. When you look at this, you will see that whether it's a man or a woman, because you will be able to identify the sex gene. All right, if it is you were to do it genetically. Okay, so that is how it looks. We good up till there? So what do we mean by homologous chromosomes? What do we mean by homologous chromosomes, and what is a homolog? No, that's haploid. But when we're talking about chromosomes, right? What do we mean by homologous? All the cells are all the chromosomes that you see in front of you on the screen here, those are homologous pairs. So what do we mean by a homologous pair?
Like the same.
Well, yeah, you could say yes. So the same. So basically, a homologous pair is just two of the same type of chromosome that appear in a pair. All right, right. So we have two chromosomes of the same of each type. All right, so there are 46, but we have 46 because we have homologous pairs, meaning that we have a pair or two of the same type or two of each type of chromosome. All right, so and one part, okay, one chromosome that belongs to the pair is called a what? We know the pair is called homologous, but one part of the pair is called a homolog. Very good. Excellent. All right. Excellent. Very good, David. All right, so let's go on to mitosis. Now, we spoke about mitosis when we did reproduction in plants and asexual reproduction and all of that tralala, right? So we're just gonna look at what exactly happens within a cell during mitosis. All right, so I'm going to show you the video first, and then we will move on to the actual processes of mitosis. But remember, mitosis produces what type of result? What type of result is produced from mitosis?
Daughter cells.
Yeah, you have daughter cells. Very good. Genetically identical cells, right? So they are the offspring cells, the daughter cells are identical to the mother cells. All right. So it's like a photocopy, but meiosis is not a photocopy. Meiosis produces different genetic offsprings. Mitosis is like a photocopy. All right. It's like you take a bunch of like you guys and you just photocopy yourself, and then hey, you end up with clones of yourself, identical, same thought process, same characteristics, same every single thing, same bad habits, same good habits, same attitude, same everything, intelligence, everything. Hey, don't be me and my DL thing. Call a photocopy machine. That is when you're listening to too much press conferences. I'm sorry. Right? Okay, fine. David, I'm sure you would prefer use the Kagebushin Jitsu coat, right? Instead of the Helsing coat. So we'll go with that. Once you understand it. All right. So look at the structure of the chromosome. All right, so this is called a homologous pair, right? As we see, two that make up the pair. So this is a whole chromosome made up of two chromatids. All right, so therefore a homolog only has one chromatid, right? So it's made up of two chromatids and they are joined together with a centromere or the center. All right. So these are what are going to be split and divided and, just reproduced cell after cell after cell. All right. So let me show you all the video of mitosis before we get into the theory of it. Where is that video? And I had put the video on all right, let's see if it'll show now. Yay.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase.
Y'all take note of the phases, huh?
An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
Alright, I have the other video to share with you as well. So we'll go through that one. Well, both of them explain mitosis. Alright, so hopefully it'll help you a little bit.
Soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
All right, so we just saw two videos that explain the whole process of mitosis. So mitosis is that process of cell division where you get identical daughter cells, all right, the same as the parent cells. So what we're going to do now, we're going to just break down the stages, all right, so that so that hopefully it could help you all as well to understand what goes on in each stage, and you'll need to know what all of the stages involved in mitosis and meiosis. Now, meiosis is a lot more, is about twice the amount of mitosis. But once you all understand mitosis, I think that meiosis may not be that bad, all right? Because at the end of mitosis, you have 2 daughter cells being produced, but at the end of meiosis, you have 4 daughter cells. So that is why it is going to be twice the amount of mitosis, all right? So we have stages, okay? So we have interphase, prophase, metaphase, all right? These are our first 3 stages. So we're going to focus on these 3 first, and then we're going to go on to the other 3. So there are about 6 stages in mitosis, all right? So when it is you're talking about cell division, first off, the cells have to prepare to divide. And it is at this stage where we get the interphase stage, all right? Inter means, it also means between or among, but it could also mean before as well, meaning that it would occur before the other stages, all right? But at this point, it is like, in between. So it's leaving that point where it's not dividing to get to the point where it is dividing. So hence the reason it's a little bit in between. All right. So it is at that point where you will be able to see the chromosomes a little bit more because they will become short and fat. And that is where you'll see it on a microscope. All right. So that is the interphase stage. It is getting ready to divide. All right. Then now prophase, all right, it is the first stage. It is called prophase because pro means before, because it occurs before all of the other stages involved, all right, naturally speaking. So here is where your chromosomes would begin to duplicate themselves, all right? So now they will consist of 2 identical chromatids that are joined at a centromere. All right, and your nucleolus, which is your nuclear envelope around, will disappear and you will have spindle fibers form as a result. All right, so at prophase stage, your chromosomes duplicate, so it's no longer 1 chromatid, it is now 2, and they will be joined together at the centromere, and then this is where now they're going to get ready in order to divide themselves. All right. At the metaphase stage, meta means adjacent. So this means that they are all lined up next to each other. Remember on the video, they spoke about like an imaginary equator. Well, that's basically what it's like, imaginary equator, right? So they're all lined up together around that imaginary line. They are next to each other. And the spindle fibers now, they actually are attached to each of the chromatid pairs. All right, so they're attached to the spindle from the middle. All right, think about it as like a little umbilical cord, right? Just being attached to the center of the chromatid, like how the umbilical cord, was enabled. Right, and then afterwards now it will disappear. Okay, so these spindle fibers are going to pull apart your chromosomes eventually. All right, so you're okay with that so far?
Yes.
All right, so you'll need to explain it to me now. First of all, tell me the first 3 stages of mitosis that we just learned, and what are the processes involved in those 3 stages?
Miss.
I'm listening.
Prophase.
Before prophase
This was before prophase.
is a phase that starts with I.
I remember phases are with I.
Mm-hmm.
Dimitri, help.
So we have the I phase.
Oh my god, like David, help. Interphase.
Very good. It's when the chromosomes will, I don't know. Dimitri won't know.
That's okay, David, help me.
So we have interphase. So
Prophase.
at least we have that part. We have interphase is phase 1.
Prophase.
What is
phase 2.
excellent? We have prophase, which
Okay,
is
somebody
phase.
else give you third phase, not me.
You're going so good. Very good. And then we have metaphase. There we go. I listed it out for you. Right. What happens at interphase stage? Interphase. At this point, we cannot see the chromosomes. So then what is going to be happening at this point? This is when the cell is, I gave you all a big hint. Before it reaches to dividing, it is getting ready. So it is preparing.
Yeah, preparing for mitosis.
Right, so it is preparing for division. D-I-V-I-S-I-O-N. You remember? Okay, so David, very good. That is metaphase stage. All right. But before we get to that, we have the prophase stage. What happens during prophase? Right. Well, not the cell divides, but what happens to the chromosomes? Now, at interphase stage, it's becoming short and thick. Chromosomes start to coil up and become short. Right. So both are correct. Chromosomes start to coil up, become short. 6 hours. So the chromosomes, yes, they get short and thick. And what else happens? Chromosomes become short and thick, and at this point, your chromosomes will begin to
separate.
Not separate yet. Before they could separate, they have to duplicate themselves. So they duplicate themselves, right? Because remember, the chromosomes themselves will have to form an identical copy, and then that copy is going to be transferred to another cell, which will become the daughter cell. All you picking Dimitri brain, boy. Oh, they're easy. But yeah, very good, everyone. So we have interphase, which is where the chromosomes become short and thick. Prophase, yes, they continue to become short and thick, but at that time they are finished and they are able to duplicate themselves. And David, take it away with metaphase. What happens during metaphase? What do the chromosomes do? If you don't want to type it, you could say it too. The metaphase chromosomes align and split apart. All right, so yes, they align. All right, then split apart, yes, but they align adjacent to each other. All right, because meta means adjacent, so they line up next to each other. at the center, all right, or at the equator of the cell. All right. So those are our three phases: interphase, prophase, metaphase. Interphase, chromosomes becoming short and thick. Prophase, they begin to duplicate themselves. Metaphase, they align adjacent to each other at the center or at the equator of the cell. The center is basically like the equator. All right. Okay. So you're good with that. Make sure to take it down because you're gonna have to recap after. So you'll get ready to fire away at your answers. Now we have these three phases, which are anaphase, early telophase, and late telophase. So just now in the video, you would have seen 4, right? Now basically the reason why these are 6 is because you have telophase 1 and telophase 2. They didn't account for that, right? But you all need to know it because first I'll start with anaphase. Anaphase will only happen after metaphase because meta means adjacent, so they're all next to each other, and then ana means up. All right. So therefore, after the chromosomes have lined up next to each other along the equator, then in anaphase, this is where the centromere split, and only here is when the fibers, the spindle fibers, will pull the chromatids at their centromeres in order to get them to the opposite end. So this is where the chromosomes will split apart and they will go to opposite ends of the cell. All right, that is what you would have seen in the video, and that could only happen after metaphase. So after they line up, they split, and that happens in anaphase. And then after that, now you have 2 end stages. So you have early telophase and late telophase. Telo means end, so hence the reason they've named it telophase, all right, because it's that phase, the phase of the end. So it is the first part of the ending. This is where your chromatids will reach the opposite ends and your spindle fibers will disintegrate, all right? And what happens is that when your spindle fibers disintegrate, your cell will begin to constrict at the equator, so it's going to begin to pinch. And not only that, the nuclear envelope as well will start to reform, all right? It's going to start to reform. But it's only when you get to late telophase that the process called cytokinesis, which you all also heard in the video, that is just basically where the cytoplasm of the cell divides, and that is how you will get your 2 separate cells, right? So it's only at late telophase you'll get that. You'll get the full formation of the nucleolus, and the whole nuclear envelope will form again. Your chromosomes go back to being long and thin and invisible, and there you have it. You have your cells completely divided until they decide to divide again. Do you all understand so far?
Yes, miss.
I am so happy to hear you say that, because now you're gonna have to teach me these 3 phases. So what are the other 3 phases we heard of just now, and what happens at each phase? So go ahead, name the 3 phases.
Okay, wait, miss, wait. Anaphase.
Uh-huh.
Telophase.
Anaphase.
Oh wait, I know you say cytokinesis, but you didn't really mention that. I don't
No,
remember the next one.
cytokinesis is a process that occurs in one of the telophase stages. So we have 2 types of telophase, which are
1 and 2.
early telophase and
Late.
exactly, early telophase and late telophase, right? So what happens during anaphase? So this will be number 4. Look at answer. What happens during anaphase? Ana means what? Remember what the actual name means, and you will remember even you will, just remember what takes place. So what does ana mean? Ooh.
Miss, anaphase is when they separate into 2 different cells, well, 2 daughter cells.
No.
Miss!
What is it? What does Anna mean? Okay, so this is, yes, exactly. The fibers will pull the chromatids to the end of the cell. Anna, as I was saying, means up. Anna means up. So for that reason, during that phase, the fibers, the spindle fibers, which are attached to the centromeres, they will pull the chromatids to either ends of the cell. So this is where they're going to be split. So the couple is going to be split, all right, because Anna means up. And after Anna, what do we have?
Telophase.
Early telophase or telophase.
Mm-hmm.
Early telophase, which is
Where they pull to the opposite side.
right. So in that case, right. So they pull to the opposite side. So they are officially pulled apart, right? They are all on opposite sides. What happens to these spindle fibers? What happens to these spindle fibers? They are, yeah. Yeah, they get destroyed or they disintegrate. Uh-huh. And what happens to the, they go boom, boom. What happens to the nuclear envelope again? It went away before, so now it's gonna, right, it's gonna be reformed. So it's starting to reform, okay? That is during early telophase. And the late telophase, this is the ending of the end. What happens during late telophase? Emily had mentioned a process before. Anybody remember that? Right, what will split apart? And what is the name of that process? Think cytoplasm.
Cytokinesis.
Excellent. Cytokinesis, which is what David said, is the splitting or the dividing of the cytoplasm, right? Which is dividing of cell cytoplasm. All right, so that's where you have cytokinesis occurring, the dividing of the cell cytoplasm, which is going to result in 2 daughter cells. Right, and that is how you will actually have your 2 cells. Hard luck. We are feminists in cell division, so you don't get son cells. You only get daughter cells. So you get daughter cells in mitosis and you get daughter cells in meiosis. That's an interesting question, though. I never thought about why they call them daughter cells and not son cells. It really is interesting, though. Because yeah, I really don't know why they call it daughter cells. I never delved into that. That's interesting. I'll research that. All right, so y'all understand the stages of mitosis?
Yes. Maybe they call it daughter cells because all babies are a girl first, right?
Well, male and then eventually, yes. Remember
Okay,
that's
well then
how,
I don't know. I was just trying to find a solution as to why.
well, understandable. But yeah, remember that's how like we are all male first and then eventually like we become female if you know the good Lord determines
I
us to
thought they
be
were
female.
all girls first and then.
Nah, it's the opposite actually. And that's how that is actually how we have a clitoris, because that is we were all forming, we were all getting a penis first and then it eventually would have, you know.
We were all boys first.
Yeah, so we were all boys at one point. Uh-huh. Yes. So, so yeah, even in gender reassignment surgeries, what they try to do is reverse that process. So, in order to, do surgery to make a male person into a female again with female genitalia, that is in essence what they will try to do with the penis again. So, let it become a clitoris, and then they will use certain tissues from different parts of the body, like your pericardium or wherever else, even a cell from inside your cheek, dependent. All right, because you want to be able to get you want it to be able to function. All right, so they will do that, and they will create an actual vagina. Um, they can have, of course, a lot of risks with it, but that is in essence what happens during gender reassignment. So yeah. Yes. David like David like, oh, oh, oh. And Dimitri catching kicks is like, y'all are all dudes. Oh gosh, but yeah, that's what happens. Oh, David, calm your mind. All right, yeah, but I'll investigate that to find out why it is they call it daughter cells. It's just to find out the whole origin of the term daughter cells. Why did they call that daughter cells? David, you're not easy. All right, so since you all understand mitosis, let's go on to the little bit more complicated one, which is meiosis. All right. Now remember, meiosis occurs in what type of cells? Meiosis is the only type of cell division that occurs in dog cells, David? In dog cells? So if I tell you that meiosis actually occurs in you, does that what do you mean doggy cells?
Insect cells, David. It's not that hard to say.
Yeah, in your sex cells, your sperm, all right, and your eggs, they divide via meiosis, right, in order to make more. But it's actually normal cells, like 46 chromosome cells, that divide first to make your it was hiding in the back of your head. So yeah, it's actually cells with 46 chromosomes that divide in order to form gametes or sex cells with 23 chromosomes. Because remember, you have to start off with a whole before you can get half. So in order to produce the half, the half number of chromosomes have to come from a whole, right? So that is why, in a sense, you will need normal cells to produce it. All right. So yeah, meiosis only occurs with sex cells because, yeah, they are responsible for the formation of gametes. All right. So I'm going to show you all the video before we get into all of this, complicated stages and whatever, the theory behind it. We're going to look at the video first, okay? So as you can see, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's about 10, yep, 10 stages, as opposed to 6 stages from before. So let's look at the video.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over that doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so that was basically your whole overall lesson on meiosis. All right, so let's just get down into the actual theory of it now that you all have some sort of visual to work with. All right, so we have our processes just as how you would have had them in mitosis, except that you have a little bit more because remember, now you have to divide again because as you saw, when it is the cells divide in meiosis first, all right, you get back the same number, all right, or actually more once it is, because you also saw that you would have 2 pairs instead of 1 pair because if it is you had 1 pair, well then naturally when they divide you'll get back the same amount, right? But because you have 2 pairs being formed now, and you would have gotten how much it is, 92 chromosomes, you have to divide until you can actually find or get the 23 chromosomes in each. All right, so that is why you have to have a second stage of division taking place after the first one. So we number the stages as was explained so that you would know which phase you are talking about. So you have early prophase, mid prophase, and late prophase. So you have 3 prophases, all right? You actually have where the chromosomes are becoming short and thick. That is the first stage of it, all right? Then the mid prophase is where you have your homologous chromosomes pairing up to form bivalents, all right? That is what you call them. And the centrioles will separate to form spindle fibers between, all right, when it is your homologous chromosomes pair up. So you will get 4 of them. Then your centrioles separate and your spindle fibers would be formed. And each of the chromosome duplicates itself. You will get 2 chromatids being joined together by a centromere. And this is where now you will have the genetic crossing over of the chromatids, all right? The nucleolus will disappear. But remember, they would, you also where like 2 chromatids were kind of looking like they holding hands and feet, all right? So they kind of join together at the top and they join together underneath. So that is basically where genetic crossing over takes place, because there you have them being reattached, you have them attaching, and then when it is they split apart, you will get genetic material, different genetic material from each of the chromatids. So this is why, remember I told you all that in reproduction, in sexual reproduction, it's like a lottery system basically. That is why I was saying it's a lottery system, because it is a whole lottery system of genes that are actually mixing, and then the result now will be totally different from the parent. All right, so you have three prophases. You have early prophase 1, mid prophase 2, mid prophase 1 and late prophase 1. All right, we still on the first set of processes, so that's why we, after prophase, you come to meta, then ana, then telo. Remember, you have to come to an end during your first stages of meiosis, and then you also have to come to another end when it is you are finishing. So you come into two ends, because you are dividing first, all right, in order to form your daughter cells, and then you're redividing again in order to get 4 cells that are different, that are genetically different from the parent. All right, so that is why you're going to have 2 stages and 2 parts of the processes. So after our prophase stages, we have metaphase. Again, this is where your bivalent, so all of the 2 pairs of chromosomes will line up on the equator of the spindle. All right, remember meta means adjacent, and as they were saying in the video, think about the M as standing for middle, because that's where they are arranged, in the middle. All right, and then in anaphase, remember the spindle fibers are attached to the chromatids, right? To the centromere, sorry. So in anaphase now, think about the A as away, or ana means up. So it's up and away, all right? Right. So after it is, now you get to anaphase. This is where the spindle fibers will pull the homologous chromosomes, all right, which are each comprised of 2 chromatids. They will all be pulled to the opposite poles of the cell. All right. And after they are pulled to the opposite poles of the cell now, then you will have the membrane, nuclear membrane being formed around the chromosomes, and the spindle fibers are going to disintegrate, and your cytoplasm will constrict again to form 2 cells. So at this point, at the end of the first stages of meiosis, you will have 2 cells being formed. All right, but each of these 2 cells now are going to have 46 chromosomes each. All right, so you already have the genetic mixing taking place, which is the genetic crossover, and this is why you had to duplicate. So remember, even in mitosis, you had the duplication of chromosomes, and that's how you ended up with 2. Here, because you are duplicating, you are multiplying 2 by 2, and that's how you get 4. So when they are now separated, you will get the same number of chromosomes, because now you will have pairs, the same pairs of each of the chromosomes, all right, homologous chromosomes. And that is what you will get by the end of the first processes of meiosis, all right? So take a moment, remember early prophase 1, mid prophase 1, late prophase 1. So early, mid, and late prophase, remember those 3. Then remember meta, ana, and telo. So you have middle, away, and end. So beginning, middle, away, and end. Remember what happens at each stage, and I'll test you all on it. All right, so you'll have a minute to do that. A minute starting now. Quick, quick, quick. All right, your minute is up. So remember it again, early prophase, mid prophase, late prophase 1, metaphase 1, anaphase 1, and telophase 1. All right, so you'll tell me the processes and what takes place in each one. What are we starting off with first?
Early prophase 1.
Excellent. So we have early prophase 1. What goes on in early prophase 1?
The chromosomes become more visible.
Chromosomes become visible.
And they shorten and thicken.
Very good. Very good. I see David said the same thing. So excellent. What comes after early prophase 1?
Mid prophase 1.
Excellent. Mid prophase 1. What happens during mid prophase 1?
They separate. The centrioles separate.
The central what?
Centrioles separate. Something to do with spindle fibers in between.
Something to do with spindle fibers. What happens? What will happen at this point to the amount of chromosomes that you have?
They pair up.
They pair up, yes. They form what? A B word. Yes, David is correct as well. Right. Very good. So both Emily and David have the correct answers. So they form bivalent. All right. Mid prophase 1. Well, David typed it, so I don't need to type it. Right. And what happens after mid prophase? What is the name of the stage?
Late prophase 1.
Excellent. What happens during late prophase?
Chromosomes duplicate itself.
Well, the chromosomes will duplicate themselves. Let's not check it back to make sure that I have it right too. No, they're right. I was correct. They duplicate before. So what will happen now? This is where they hold hands and feet. They look like they hold hands and feet.
They join together.
You have.
Miss, I tried to remember when you turn it back.
Yeah, they join together, and this is where you have genetic crossing taking place. All right, so when it looks like they're holding hands and feet, that is where you have genetic crossing, because at the point at which both chromatids touch, they mix genetic material at that point. All right, so you would have your genetic crossing taking place there. Excellent. After late prophase 1, what do you have?
Metaphase 1.
So you
where
have
the membrane disappears.
So the nuclear membrane disappears, forming what?
Miss.
What forms in the absence of the nuclear membrane? Very good, David.
David, help him out. Dimitri is silent.
Very good. So the chromosomes line up adjacently. Nuclear membrane, and just as Emily, your answer was correct as well. You were going good. So the next thing you had to say, Emily, would have been where the chromosomes line up adjacent to each other. Right? I'm just typing it up here for you, right? So chromosomes line up adjacently, right? Anaphase 1 is next. What goes on during anaphase 1? Remember, ana means?
The spindle fibers pull to the opposite poles of the cell.
Pull who?
Pull
What does
chromosomes.
it pull? Yes, very
Dimitri's
good.
answer.
Yes, very good, very good. The spindle fibers pull chromosomes to opposite ends of cell because ana means up, up, and away. So it pulls the chromosomes to the opposite ends of cells. Very good. What happens after anaphase?
Telophase 1. Telophase 1.
Right, so we have telophase 1. What goes on during telophase 1?
Where the
Very good, David.
spindle fibers dissolve, disintegrate, I think you said, on the
Yeah.
last one, right? They disintegrate, and the something to do with the nucleus. The nuclear envelope is formed.
Exactly. Yeah, it is what David said. The nuclear envelope is formed. And what happens? The same process that you had called before that starts with C, ends with esis.
Cytokinesis.
Right. So here, because it's at a telophase stage as well, we have cytokinesis. K-I-N-E-S-I-S taken place. So therefore, that is what will separate the cytoplasm, and you have two separate cells forming. But we don't end there, all right? But before we get onto the second part, you all understand the first part?
Yes, miss.
Okay, so once you all understand, I know Dimitri is a lot. I know. Sorry, I think I could smell all the overheating brain cells from here. I know. That's why I wanted to take my time with it so we could, you know, chew it and digest it. And then even after, you all could go through it and, you know, just make it yours to understand. All right? Okay, so now we get to the second stages, which is just basically 4 of them. All right? So we have now, it has to start again from prophase 2, but it's not the same as prophase 1. Metaphase 2, then anaphase, telophase 2 again, all right? Because now we wanted to divide to get the number of chromosomes that we want. So we have prophase 2, where centrioles will separate and they will form 2 new spindles at right angles to the first. All right, so right angles to each other, okay, facing each other. So this would be our prophase 2. See how it's different to prophase 1? Then in metaphase 2, your nuclear membranes again will disappear, and your chromosomes, which are each comprised of 2 chromatids, line up on the equator again. So at this stage, we have 46 chromosomes, but now when they split, we're going to end up with daughter cells only having 1 chromatid. All right? And then when it's dividing again, it will form 2, and it will duplicate, and the whole process will happen again. All right? So at this point, we have 46. So now when they split now, each one of them going to have 23 chromosomes. So at anaphase, when the centromere split and the spindle fibers pull the chromatids to opposite poles of the cell, that is where it will pull apart the 46 chromosomes, and you will get 23 and 23 on both cells, on all cells, right? So after that now, at telophase 2, which is your final end stage, your chromatids would reach opposite poles of the cell, your spindle fibers disintegrate, your nuclear membrane forms again, all right? And the compositions of your chromosomes would be half, all right? And you will get, in total, 4 genetically different daughter cells. All right, so this is prophase 2, metaphase 2, anaphase 2, and telophase 2. So these are the final stages of cell division in meiosis. Yeah, I feel you, David. It is a lot. You'll want to look at the video again before we try to review the processes like we were just doing. All right, let me just show the video again then. Oh, I took it off.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle, The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. So look, I'll give you all 2 minutes to just remember these, and then we'll just do a little quiz on them. All right. Prophase 2 to telophase 2, and what happens at least in the latter parts of it. All right, so let's go. We're just dealing with the second phases, right? The second stages. So remember, prophase 2, we have the centrioles separating. You get two new spindles again, forming right angles to each other. Then metaphase, the nuclear membranes have to disappear and your chromosomes line up again on the equators. Then ana is up, up, and away. So this is where the spindle fibers will split and pull the chromatids to opposite poles of the cells. And your telophase 2, which is where you will have your spindle fibers disintegrating because the chromatids would have reached opposite poles of the cells. And once they are, in those opposite positions, you will have the nuclear envelope beginning to split again, and you will get new cells, all right? And of course, the compositions of the chromosomes would be different, all right? So remember these, so let's go. Prophase, metaphase, anaphase, and telophase. So Tama, where's the first step in phase 2? David, pass on it, boy. Prepare that. Centrioles separate and form spindle fiber. Very good. Prophase 2. What comes after prophase 2?
Metaphase 2, where the nuclear membranes disappear.
Very good. Nuclear membranes disappear and very good, Cheyenne. That's okay. What happens during metaphase?
The chromosomes line up on the equators of these spindles.
Precisely. Very good.
Chromatids or
Right.
chromosomes.
The chromosomes line up, yeah. The chromosomes line up. It's the spindle fibers that would be attached to the centromeres, right? But it's the chromosomes themselves that would be lined up in the middle. So very good. So that's metaphase 2. What is the next phase? Excellent, excellent, Cheyenne and David. I like it. So we have anaphase 2. And yes, David. Right. What happens in anaphase 2? Remember, this is your up, up, and away state. So what happens here?
Where the chromatomes pull to the opposite side of the cell.
Yes, centromere
Opposite
split.
poles of the
and-
cell.
Right. The chromatids are pulled to opposite ends of the cell. Very good. Excellent. What comes after anaphase 2? Very good. Telophase 2. That's okay, Cheyenne. I appreciate your response still and excellent work. I prefer to receive it regardless of what. All right. Ow. So that's very good. What happens in telophase 2? The final end, the end of the end. Chromosomes are now at two poles of the spindles. Nuclear membrane forms at each chromosome. Very good, Cheyenne. And will be split apart and a new. Very good. That's some type where you have there, David. Yep, very good, both of you. And then what else will happen so that we get separate cells now? The C word that ends with esis.
Cytokinesis.
Very good. So remember cytokinesis because it's where the cytoplasm splits. So cyto and cyto. All right. So yes, very good. Your cytoplasm will split, giving you new cells. And what's the number of chromosomes by the time meiosis is finished? You started off with 46. Very good, 23. So you started off with 46. You eventually get to actually 92, and then you get back down, all right, to 46, and eventually you will get 23. All right, because when the chromosomes duplicate, because you're starting off with 46, when they duplicate, that's why you get 92, and then they split apart at the end of telophase 1. When they split apart, you will get 46, and then at the end of telophase 2 is when you will get 23. So that is actually the number differences that occurs. So your body is very good at mathematics and dividing, unfortunately. I mean, fortunately. Okay. So the next part that, yes, in the cells, your body cells. Yeah, I could attest to that too, okay? Because I cannot visualize properly. I have to write down and, you know. So yeah, maths and me, we're not that good friends. All right, so very good. Maths is a different level. You're right about that. Okay, so I'll give you all a break from the mitosis and the meiosis. Once you all are okay with that, we'll just go on to the other stuff that I wanted to do. I'll just briefly talk about stem cells. So the stem cells, I was talking about when we were doing reproduction and the baby formation and all of that. Remember, we were talking about how cells, they, before they get like a particular function, they are just there functionless. Okay, so basically, until you have functions assigned to a cell, they can become anything. All right, and what happens is that your proteins are, once you have protein formation, that is what will determine the how you call it, the function of a cell. All right, now what happens is that as a baby is developing, your cells will begin to take on different roles, because I mean, you will need cells to that will be responsible for performing every single thing in your body. All right, so when you talk about cellular differentiation, we are referring to that process by where a cell gets a role. So this is where your cells get jobs, they are employed. Unfortunately, they don't get money, so it's not like you're gonna have money floating around on the inside of you, which kind of sucks. It would be cool if you could have withdraw cash from your body. And put, like how in SpongeBob, Mr. Krabs was like an ATM. And I think anytime his whale daughter wanted money, it just came out of his chest basically. So yeah. Unfortunately, we're not like that, but yeah, they do get a job, they do get functions and rules. All right. Plankton, I really don't know. Plankton's son is an ATM, but Plankton, how? I had to find out. Thank you very much. I try. I really didn't know Plankton's son is like an ATM. I thought Plankton was just a loser. Right? So what happened is that even though you remember I was saying that you have the same amount of the same amount of chromosomes in each cell, right? And you have your same genes. So as I say, the same gene that is responsible for growing hairs in your nose hole is the same gene you have in your foot or in your arm or on your face. However, it is not activated anywhere else, but it is activated where it's supposed to be. All right, so the genes can be switched on or switched off. So if a gene is switched on, then basically the particular gene, which is the particular instruction, will be relayed to the cell, and that will determine what the cell moves on to do, okay, and how it is expressed. If a particular gene is switched off, then it will not have that process, all right? It will not function in that capacity then, all right? So the gene for nose hair growing, right, is switched off anywhere else in your body except inside your nose. All right, so that is where we get all of that. All right, so that's just briefly with stem cells. But the part that I actually want to go on to is where we come to inheritance and characteristics, all right? So on your chromosomes, you have your genes, right? You have your chromatids here, your centromere, and you have, for example, genes that are responsible for your eye color that might be on this part of your gene. Genes that are responsible for your hair color might be somewhere just underneath here. So all of the genes that are responsible for different parts of your characteristics are found somewhere on the chromosome. All right? So this brings us to talking about what is an allele. So this is where things get a little bit more complicated, but once you'll understand this, you will be good. All right? So a gene is just basically instructions, all right? They are responsible for producing or forming a particular type of protein which will determine characteristic and cell function, all right? So what happened is that, like, for example, in chinchilla rodents, which are basically rats, all right, you have like genes that determine the color of the fur. Even in your animals, like your dog, your cat, your duck, if anybody have a duck, or your chicken, all right? So genes are what are responsible for determining that color, all right? And your genes will produce the proteins that will cause different fur color, because what will be responsible for the colors are pigments, and pigments would be determined by proteins, all right? So that is what a gene is, just basically a set of instructions responsible for producing proteins that determine characteristics, or they are just instructions, period. But then now you have an allele, which is just another form of a gene. So what happened is that while you have one gene, but there is another way in which that gene could be expressed. So an allele is just a form of a gene, another form of a gene. All right. So like, for example, what happened is that in order for you to express these alleles or different forms of a gene, which is just like characteristic, okay, a different form of the characteristic, you can use symbols, right? And so like if it is you wanted to talk about the fur color for a chinchilla, all right, and you wanted to talk about the fur color being gray, to represent that fur color, to show that it's an allele, you can use capital G, because that is said to be the type of the more expressive gene, all right? We'll get to the terminologies in a little bit. And what happened is that you can use common G to represent the allele or that particular form of gene that will not produce gray, but it will produce charcoal fur color. So you see the capital G and the common G, those are genes, right? But they are alleles, or they are alternative forms of that one main color, the gray gene, because you can either get gray or you will get charcoal. All right. So basically, the main gene or the main allele that we are speaking about, which is actually what we can see, all right, is always use we always use a capital letter, and the common letter is what we use to get the secondary characteristic, all right, what we may not get, or what might just be lingering inside of you but may not even be showing. So let's just say, for example, I have a baby and my baby has blue eyes, all right? I don't know how he has blue eyes, but he come up with blue eyes. I don't have blue eyes, my husband don't have blue eyes, but my baby come up with blue eyes, all right? No, nothing happened. I was completely faithful, everything was good, but my child had blue eyes, all right? That is because the gene for eye color, or that particular allele, all right, for eye color was actually somewhere in my genetic history. And then only at that point where you had genetic crossing taking place, that switch and, you know, little shuffle up the lottery system, the blue gene for eye color, the blue eye color gene came out, and that was actually expressed in my baby. But it doesn't mean to say that my baby have no capability of having a child with brown eye color. It just means that primarily, all right, it has blue eyes. So that means that the color for blue eye would be capital because it's what you can see. It is dominant, we will see. All right. And because we cannot see the brown eye, but it's still there in him, in his genes, we will refer to that as being the common color because it is what we call recessive. All right. And then when my child has another child, that child could actually have brown color eyes. All right. I know this is a little bit tricky, but just bear with me. All right. Hopefully it will start to make sense in a little bit. All right. So you can have different allele combinations, which is different forms of genes combined in order to produce certain results. So you can have two capitals, a capital and a common, or two commons. All right. This will just be the other end of the spectrum. All right. And this is what we start off with to determine how your offspring going to come out. So what happens is that you can have possibilities by once it is you know what the parents are, you can have different possibilities for your offspring. So if, let's just say, for example, the parent has, like one parent has, all dominant gray genes, and then the next parent has, yeah, dominant, but it also have the capability of, having charcoal fur. When both of them come together, you will get different probabilities because you will have this allele mixing with this allele and so forth. All right, so you could end up with either a rat that has all gray fur or a rat that has, not all gray, but it actually has charcoal. Or even though if it has the main gray, it will have the possibility of getting charcoal fur. All right, let's just get to the definitions a bit. Right. So you have homozygous and heterozygous. All right. So homozygous means that you have the same particular allele. All right. So you have two alleles for a particular gene in a cell that are being the same. So homozygous is just when two letters are the same. Whether they be capital, whether they be common, they are homo, they are the same. All right, just how you get homosexual, meaning same sex, homozygous means the same. All right, allele. Hetero means different. So therefore, just as how heterosexual refers to different sex, like man and woman, right? Heterozygous mean different. So therefore, the alleles would be different. So a heterozygous allele will be capital and common. So here we just basically going to look at how characteristics are mixed in order to produce different results. All right. You see what it is, you see the results that you get are based on these. All right. These alleles and their combination mixture, these are what you get. So capital G will be homozygous. Capital G, common G will be heterozygous because they are different. Common G will be homozygous. All right. The characteristic that you see means that the gene responsible for that characteristic will be dominant. All right, because it is dominant because it is that characteristic that you see. So if I am born with brown eyes, it means that my gene for brown eyes had to have been dominant somewhere. All right. So even though I may have the possibility of having blue eyes, what you mainly see is brown eyes. So that means the brown eye gene is actually dominant, but I am recessive for the blue-eyed gene. So even though because it's recessive, it's just hiding away in the background now. So because it is there in the background, you will not see it, but somewhere in my future generations, this will be expressed. All right. And on the contrary, common G in this case is referring to charcoal fur, all right, because it is the opposite of the spectrum. So we're not dealing with gray. Gray is capital G, charcoal is common G. So in this case, if the offspring has common G, it means that it is charcoal. All right, so this is homozygous, yes, but it is recessive. So this is where we get to genotype, phenotype. Oh, here it is. Dominant and recessive. All right. So dominant, it is the expressed gene or allele, sorry, that once it is present. And recessive is only expressed when you have no dominant allele. So when you don't have any dominant allele and you just have common letters or common alleles, that is when you will have recessive alleles coming into play, and that is when you will see the characteristics of it. All right. So what happened? What is what the other two definitions that you all need to know are genotype and phenotype. The genotype, look, you have gene, all right, meaning type of gene. So genotype has to do with the gene. So it's what the organism possesses in terms of the genetic makeup. Phenotype is physical, all right, so the visible features of an organism or what you are actually able to see, all right? Even what it is you aren't able to see, it's just basically the result of the reproduction, basically, all right? What you will have, all right? How y'all going so far? Y'all following with me so far, a little bit?
Yeah, Miss, we did this in HSB, so I still have my HSB notes to follow from as well.
Wonderful, wonderful. Yeah, Dimitri, it is a bit difficult, all right? This is the most difficult topic, I think, in bio. But after this, it gets really nice. So I'll show you all how we could actually calculate this. All right. So let's see. All right, so we looked at dominant and recessive. So while you can have dominant characteristic, dominant meaning that the allele is dominant, okay? That is what you can see. All right, now remember, you cannot have when it is you have a dominant and a recessive coming together, as in capital and common, the common being the recessive allele could never take precedence over the capital, never. Dominant will always remain dominant. Once dominant allele is there, that is what will show. The recessive will remain in the background, right? But dominant will always take precedence. However, in the absence of a dominant allele, and you have two recessive or two common letters, then this characteristic only will then be shown. All right? So that is how this whole thing works. So just remember that. But even though you have dominant and recessive, you could have an instance where you have co-dominance, where you don't have recessive, you just have two leaders who want to lead. So if it is you're talking about color, when you have two leading characteristics, they will merge their characteristics. So here in the flower, our phenotype, or that which you can see, is red, right? But the genotype, all right, is C, which is for the color, all right? C, R, which is red, and R, red. It's capital because it is dominant, and they normally choose a color based on, you know, what it is you're dealing with, all right? Also, you would have white, all right? So you have dominant color being white, dominant white. So here we have dominant alleles. However, if two of them are crossed, you will get a mixture of these two colors because you are dealing with dominant colors, all right? Now this is what could happen in plants, all right? So you have a dominant red with dominant white coming together to form pink. And both of them are dominant. So you get color white with color red. Both of them are dominant. Red and white produces pink. And voila, you get pink. Do you all understand that a little bit?
Yes, Miss.
Wonderful. Dimitri, David, Cheyenne, how are we doing?
I like this topic actually. I find it easy.
It's cool. It is cool. Really, really
And
cool.
I know there was a way to find out, like if you have two people and you have their allele, you could use the square thing to figure out. We did it in HSB, but I couldn't remember what it was called.
Yes, it's called genetic diagrams, which I want to get to shortly. Right?
Yeah, I had like that one. So you could see what, like how many, like if they have a child, what their like if they have brown eyes or blue eyes, the same example.
Exactly.
We'll see which one will get brown eyes and which one will get blue eyes.
Exactly. Exactly. So we could actually work that out. So when I teach you how to do it, we could work out the brown and the blue eyes, right? Which would be like capital B, common B. All right. So once you're good, let's do. Now, in the case with blood, like ABO blood group, what will happen is that you have three alleles which govern, three alleles of that gene which govern this, right? So, which would be you have A, B, and you have B, all right? But what happened is that if a person, all right, with the genotype, well, IA or IB, if they were to, merge and you end up getting an offspring, you could have AB blood group, all right? Characteristics of both A and B will be shown, all right? But you see, if it is a person has ABO blood group, you will have characteristics of all right? But A and B will be predominantly exhibited, all right? So yes, that this is also another example of codominance, just when it is you're dealing with blood, all right? As you can see, all of them will be dominant. However, if anybody has ABO, the A and B will be dominant to O, all right? If you're dealing with all three. That's how it works, all right? But yeah, you will have antigens from all of them and you will exhibit properties from each of them, okay?
Miss, my mother has O positive and my dad is AB positive, but Sanjay and I are both A positive. That's what happened in our case.
Well, in all the case, y'all, it's not to say like y'all came out, okay, so both of y'all are A positive, right? And your mom
Yeah,
is O positive and your dad is what?
AB.
AB, right? So in all your case, it didn't have like a combining of both of them. So you're just inherited parts. So none of y'all inherited your mother's blood type. You're just inherited basically your father's blood type.
Part of that. Okay, yeah.
Yeah. So, oh gosh, Ollie, you mean some y'all don't have Ollie can't save all your mother? Way boy.
Oh,
Oh
none
my
of us could give her blood.
No, so you mean to tell me your mother could save all you and Ollie
But
can't save
we
all your
can't
mother?
save her.
Oh my gosh. Wow, but it's really interesting, huh? But yeah, that's how it would have been. So the scale just tipped in that favor.
Yep, it's in dad's favor.
Pretty much. All right, let me see if I have anything here to explain. Right, now in gametes, what happened is that because you have half and half, you have 23 chromosomes in sperms, 23 chromosomes in egg, what would happen is that your those cells could only carry one pair of the alleles for the genes, right? So like for example, if like in a male rat or chinchilla, if it's heterozygous, meaning that it has one dominant, one recessive, it is a carrier for the charcoal fur, right? So in its testes, when the sperm are made by meiosis, all right, you will have a sperm cell, the sperm cell getting either the capital G or the common G allele. So it will get either this or this. It wouldn't get both. So therefore, when the sperm mixes with the egg, the batch of sperm that, it could either be the batch of sperm that has the gray dominant allele mixing with the egg, or the sperm that has the recessive gene for charcoal fur mixing with the egg. So it's either or. So this is why it's a lottery system that happens. All right. So half of the sperm cells will have genotype G and half will have genotype common G. If it were capital, then half would have capital, half would have capital. All of them would just have capital. All right. If it were both commons, then half would be common and half would be common. All right. The whole thing would just be recessive in general. All right. So with that in mind, let's just go to, I want to explain the genetic diagrams before I let you go. All right. So sorry, this is what I was just explaining. Each sperm cell would receive either one of the alleles, right? So half will have one part and half will have the other. All right. But for example, if a female has like for example charcoal fur, then that would mean that she only has, her genotype will be GG, both recessive, right? Because that is the gene responsible for producing charcoal fur. But that will mean that all of her egg cells will carry G allele only. All right. So, but if it is she were heterozygous, meaning that she had both capital G for gray fur and common G, well then her egg cells as well will either be capital G or common G. All right. So it's whichever egg decides to mix with whichever sperm. All right. And that's what will determine your outcome. So let's go on to learn how to determine an outcome. So this is where you can use a genetic diagram to calculate possible genotype and phenotype of an offspring. So you can find out the probability that you would have, like what genes your child might be born with and how your child might look by using this once you know your particular genes, all right? So with a rat, for example, the first thing that you need to do is write down the phenotype, which is what you can see. So we're dealing with a total gray rat and we're dealing with a charcoal rat, all right? So gray and charcoal. These are the parents' phenotypes. Then the genotypes that you will get, for in this case, we're dealing with heterozygous, right? We're dealing with capital G, common G. All right. And the charcoal, well, as we just said, charcoal will only be common G, all right, because this is the recessive allele, all right? When the cells divide in the testes, because we're dealing with male first, you will get either capital or common G being produced. So you must put your gametes as being either capital or common. For your charcoal, it is only one type of gene that is, being expressed. So therefore, you will only get one type of gamete being produced, which is charcoal food. You put that in a diagram. You put your male on the left side, and on top the female goes. So the sperm goes to the left, egg goes to on top. You put down your gamete for your sperm, which is capital G common, capital or common, and then you put this is supposed to be recessive, a common G, all right? They made a mistake. And then you multiply. So capital by common will give you capital and common, all right? Common will never go before capital. It's always capital and common, all right? And then you have common, multiply by common to give you two commons, which is, this is heterozygous and this is homozygous, recessive. All right, so you can either get offspring 50-50. As you can see, it's half and half as demonstrated by the little box. So you can either get, there's a 50% chance that the offspring will come out heterozygous for gray fur or homozygous recessive for charcoal fur. You understand how to do this?
Yes, miss.
Anybody else? No, this one is the easier one. We're going on to the one with 4 so far. Oh, well, in a little bit. Anybody has any doubts so far? Oh, okay. Where? Okay, tell me, Dimitri, where did we lose you? Is a lot. Okay, so at least, okay, so here what I'll do, right? We'll explain, we'll go through. I have a question for us to work through. So after all of this, you know, when it is your brain a little bit less saturated, I know it's a lot to take in at once. Yeah, it really is plenty. So at least that's why you will have the recordings to go through it, and I will send you all practice as well, so that you can do the practice and send it to me. All right. And yeah, I finished correcting everything else, so therefore I will be able to give you all feedback in time as well. So you can send me the practice. I have to just source up a little bit more with the genetic diagram, so you all could practice more of this. All right. So let's just okay, so let's do one like this. This one is easier. So let's go. Let's just say, isn't there a whiteboard?
If only we had one more class.
Oh, I
So
know.
it could have been easier on us.
I know. Where's the whiteboard? Didn't I have a whiteboard? That's the annotate. Why am I not seeing the whiteboard option? All right. Not seeing the white. Oh, let me just stop the share for a while and see if I can get the whiteboard. Nope. Now we can. Oh wait, there's a whiteboard here. Sorry. Right, you're seeing the whiteboard?
Yes, miss.
All right, good.
We could even write on it.
Awesome. All right, so let's just say, let's deal with the phenotype, right? Remember, phenotype, I'm not writing the whole thing, right? Phenotype is what you can see, the physical, all right? So let's just say blue eyes or blue color. And we do, so if blue is the dominant one, right, first, we using, let's just say, something that is non-blue. So we'll use brown, blue or brown, right? Oh gosh, my handwriting crackle foot. Right, so then after your phenotypes, you write down your genotypes, which will be your genes responsible for this, right? So let's just say that the blue is homozygous. So therefore, we will get, if it's homozygous, capital, because it's homozygous and this is the dominant color, all right, that we're dealing with. Brown is not blue. So therefore, if we are mainly dealing with capital B, anything other than that will be a common letter to this. So that's why we will get common B and common B. All right. So now if these two were to meet, let's look at the gametes that we're going to have with these two. You can either have capital B or capital B, all right? So all of them will produce capital B because it is the same type of allele. If it were capital common, then you will get either capital or common. But here we're dealing with two of the same. So for that reason, it could be capital or capital. All of them will just be capital. And for the egg now, it will only be common, all right? So when we draw our little table to work out probability now, oh gosh, here we put in our sperm, here we put in our egg. So our sperm has capital B, and our egg will only produce one because remember sperm mating with the egg, right? So the egg will only produce one type, again B, all right? So in this case, since both of them will mate, multiply capital B by common B and you will get capital common or capital common. So in this case, you all will see that all of the offspring, there is a 100% possibility that all of the offspring will come out heterozygous for the blue eye or blue fur characteristic, all right? Because it's capital and common. They are heterozygous, but all of them will look blue because that is the dominant gene. So they will all be blue. You all see that?
Yes, miss.
Okay. I see somebody message. Wonderful. All right. Let me erase this now. Erase. Ooh. Why I couldn't just play it for you automatically? Why we had to do all that work? Okay. So let's now look at another one. So we're dealing with phenotype and we're dealing with genotype. So when you write it out like this, it actually makes it easier for you, right? And then we're dealing with gametes before we get into anything else, right? One more pen. One more pencil on. I don't like this. Oh, okay. Sorry. All right, so let's tackle a different type now. There we went with one type, but if it is you had two heterozygous parents, what would you do? So let's see, let's take the chinchilla. Let's go easy, right? We were already talking about the chinchilla, which is your rat. So let's go with heterozygous parents. So we go in with capital G. Oh, sorry. Here we write in oh gosh. Here we write in phenotype.
Miss, you're on eraser.
I know, I'm trying to go back to, I don't like this. This thing giving me trouble. Okay. All right, good. We'll be back to normal. All right, so we write in phenotype, right? I'll just write P for phenotype to save on time. We're looking at gray. But both of them, let's look at gray because we want to go with heterozygous, right? So we're going with gray. So this is your phenotype. This is your genotype. So we're looking at heterozygous, right? So both genes, both alleles have to be different. So we need a dominant and we need a recessive. We need a dominant and we need a recessive because we're dealing with gray. Okay, let's just say two of them, they are both dominantly gray, but with them being heterozygous means that they are carriers for charcoal fur. So what's going to happen to their offspring? That is what we're going to see. Let's look at what will happen for our gametes, right? For our gametes, because we have two different alleles, heterozygous, you can either get, you'll get a batch of sperm cells that have capital, that will have the gray allele, or half of the sperm cells that will have the charcoal allele. With the egg as well, you can either have a gray produced or a G produced. All right, it depends on which egg is released at the time of fertilization. All right, whether it be an egg with the gray allele or an egg with the charcoal allele. Okay, but now for this one, we need to do our little diagram a little bit different because we're dealing with two heterozygous parents, right? So here, because we have 4, 1, 2, 3, 4, we need 1, 2, 3, 4. Our sperm goes here and our egg goes here. So the sperm, the gametes for the sperm will be capital, common, always capital first. And then for the egg as well, capital and common. And now we just cross multiplying, and the result that we get here will determine what are the probabilities of the offspring that we will get. All right, so you can have capital G by capital G will give you GG, all right, which is homozygous dominant. Capital G common G will give you heterozygous for gray. Capital and common, again, capital and common. And then common with common will give you common. Remember, when you have a capital and a common, the common will never overtake the capital because the capital is dominant. So dominant always takes dominance, right? So according to this, you have, now remember, each square here would represent like 25%, right? Because 25 by 4 is 100. So you will have a 25% chance of having a offspring that is homozygous for gray, capital G, but you have a 50% chance of producing an offspring that is predominantly gray, yes, but heterozygous. So if it's heterozygous but predominantly gray, it means that you have a capital G allele and a common G allele, all right? But then you have a 25% chance also of having charcoal offspring. So this is the probability that could occur when a male and a female rat mate and one has, and both of them are heterozygous for gray. So this is how, in essence, you read your genetic diagram as well. You cross multiply the letters and then you go by your percentage probability. So 1 square represents 25%, so that's how I get 25% chance of them having a homozygous dominant gray, or 50% because you have 2 squares with heterozygous dominant, or 1 square, which is 25% of having a charcoal homozygous gene. You all understand that? You all see that?
Yes, miss.
Wonderful. How about everybody else? Wonderful. So this is in essence what the whole genetic diagram hullabaloo is. All right. So yeah, that is, yeah, that's basically it for that. All right. So I don't think I have any reason to bust out the brain again. Well, it have other stuff on sex linkage and all of that, but this is the main part that I wanted to get to, basically. All right. Okay, so how are you all so far, besides oversaturated? You all okay? You think that you'll listen to the audio and you'll be able to, eventually work it through again? And I'll send you all the practice as well.
Yes, miss. Because I did it in HSV, it just kind of is like more revision and like I understand it better. So
Very good. So before you all go, there's this, let's just do this question. Okay, so first of all, what y'all think is the answer for number 31? So the diagram here shows a diploid cell and alleles capital R, common R. So it is it what? A homozygous or heterozygous?
heterozygous.
Very good. And this is in one pair of chromosomes, right? So when this cell now divides by mitosis, which daughter cells will be produced? What y'all think is the answer? What will be the chromosome number if it divides by mitosis? What will be the genotype if it divides by mitosis?
A
No.
would be the answer. Diploid
Excellent.
and heterozygous.
Excellent. So I was not going to say know the type of cell division, mitosis. So therefore, it's a diploid cell. It is dividing by mitosis. So the chromosome number will be diploid. It will remain the same because mitosis produces the same number of chromosomes. And because we have a dominant and a recessive pair of alleles, it is called heterozygous. So the same type of, the same pair of chromosomes, heterozygous, will result in the genotype. So that's how we get A and, well, sorry, diploid and heterozygous. Very good. And the next one. So the diagram there shows breeding rats where the allele for gray fur is dominant to white fur. All right. So we could use the allele fur, they said gray fur is dominant to white fur. So therefore, what are your alleles then? Tell me the numbers. If gray fur is dominant to white fur, all right? In this case, we could use capital and, well, of course, we're using capital and common, all right? We could use common W for white and G for dominant. So tell me, what will the alleles be? If, what could the possible alleles be? Gray fur is dominant to white fur.
So it could be capital G and lowercase w.
Very good. Capital G and lowercase w. So now we want to know which two individuals are definitely heterozygous for the fur color. So therefore, which two of them that would be heterozygous to produce the heterozygous, which two of them would be heterozygous?
2 and 5. This kind of confusing in the numbering. I'm not sure what is what, like when they say 1 and 4.
Oh, okay. 1 and 2 are two parents that will come together to form these two rats with these two colors, right? And 3 and 4 are the two other parents that will produce these type of rats. So they want to know which two of them are definitely heterozygous for the fur color.
2 and 3.
No, it wouldn't be. Okay,
Oh,
3
wait.
is correct. 3 is 1, but it wouldn't
3
be
and
2.
6.
Excellent.
3 and 6.
It would be 3 and 6 because look at the outcome. It wouldn't be 1 and 2 because for you to get all gray like this, It will mean that this is definitely
It would just be
dominant.
2 capital G.
Exactly, this is dominant, right? And it could also mean that this one is how you call it? Recessive, not recessive, sorry, it could mean dominant as well, or it could be heterozygous, all right, because they did say that gray fur is dominant to white fur. So even if this has some form of it, all right, it wouldn't show. But because of the fact that you have gray and gray, it means that you can't have heterozygous here, all right? But the two that are definitely heterozygous will be 3 and 6, because out of these two, you could have a probability here of it being heterozygous because you have a white and a gray. But because you have 2 gray here, it means that, yeah, this definitely cannot be heterozygous. Understand?
Yes, miss.
All right. Who's that?
Oh, that was Sanjay in the corner.
Oh,
He in English class next to me.
oh, nice. All right, y'all. So with that, I will let you all go. All of this information was sent to you, and this class recording is on the cloud of Zoom. I think it's being recorded on the cloud. So yeah, it will be accessed and posted on YouTube for your access as well. So you'll be able to, go over and get the practice and stuff. All right. Yes, it's on the Zoom cloud. All right, so y'all, sorry that the last class had to be so frustrating, but thank you all very much for being a part. Thank you for your continued dedication, for your support, for everything basically. Just thank you for being a wonderful class. I thoroughly enjoy teaching all of you. You each brought your unique personalities, individualities, and own vibe to the classroom that made it really wonderful and fun and even encouraging for me to, just do better to teach you all every time. I hope that I did your justice, and if not, I really do apologize, and I will continue to improve.
No, miss, you do way more.
Oh, thank you. Thank you. So sweet. So it really has been such a pleasure. And like I said, this is not the end. You all could continue to reach out to me, and just anything at all, you could feel free to message, and I'll help you all out in any way that I can. All right. So you all take care. I wish you all the best and everything.
You
And
too, miss.
thank you. And well, Emily, I'll be seeing you in my online class on.
Yes, you wouldn't miss me.
Oh, wonderful. So I'll be seeing you on Thursday, and the rest of y'all, I'm sure I'll see you all again. All right. So bye. Take care. Y'all be safe. And yes, for the foreseeable future. So y'all be safe. Continue to do what you need to do to be safe from all of this COVID stuff. And I pray everybody and your families to be safe as well. And don't worry, this too shall pass. So we'll be good and we'll get through. All right. So take care. Have a wonderful night and all the best and everything.
Bye,
And
miss.
bye bye. Take care.